Welcome to today's Explainer. I am literally so thrilled to guide you through this fascinating material today. So, I want you to think of the human body as this incredibly complex machine, right? And it's built and run using a highly detailed instruction manual. That manual is our DNA. But what happens when there's a typo in those instructions? For basically all of human history, we've only ever been able to treat the symptoms caused by those typos. Well, today, we're looking at a modern medical biotechnology that changes all of that. We're talking about gene therapy, our actual modern ability to go in and fix the typos in our genetic code. It is nothing short of architecting the genome, and the science behind it is just incredible. Here's our roadmap for today's Explainer. We'll be looking at editing the human blueprint, how gene therapy works, delivering the genetic payload, curing the incurable, the promise and peril, and finally, the future of medicine. Okay, let's dive into this. Section 1. Editing the human blueprint. Take a second and just imagine a world where we don't just treat the symptoms of an illness, but completely cure genetic diseases at their absolute source. For decades, medicine is largely focused on managing disease, right? You take a pill to lower your blood pressure, or maybe you use an inhaler to open up your airways. But what if the treatment wasn't just managing the downstream effects? What if it was correcting the foundational code that caused the issue in the very first place? That is the core question driving this entire field. So what is gene therapy exactly? Basically, it's a biotechnology approach meant to replace or repair faulty genes. Returning to our instruction manual analogy for a second, gene therapy is the process of finding the specific page with the critical error and either introducing the correct paragraph, taking out the faulty sentence entirely, or altering specific words so the body can function properly again. It's not a drug you take every day. It's a fundamental modification of your genetic material to correct disease-causing defects. Now, with that definition in mind, we really have to look at the two fundamentally different ways this can be applied. First up is somatic gene therapy. This targets non-reproductive body cells. And the absolutely crucial point here is that it only affects the treated individual. If you receive somatic gene therapy, those changes stop with you. They are not passed on to your kids. This is the most commonly used form today. On the flip side, we have germline gene therapy. This targets reproductive cells, like sperm, eggs, or embryos. Because of this, the changes are heritable, meaning any genetic edit will be passed down to future generations. And as you can probably guess, and as the source material specifically notes, this makes germline therapy incredibly controversial ethically, and it's mostly restricted or outright banned in many countries right now. Moving right along to Section 2, how gene therapy works. There are three main mechanical methods of gene therapy you need to know. First, we have gene addition. Think of this as taking a pen and simply writing a brand new functional gene right into the manual to take over the job of a defective one. Second, we have gene editing. This is more like using a highly advanced word processor. Using modern technologies like CRISPR-Cas9, scientists can actually go into the DNA sequence and make incredibly precise modifications. And third, there's gene silencing. If a faulty gene is actively causing harm, we can use RNA-based techniques to act kind of like whiteout, effectively turning off or silencing that harmful code so it can't cause disease anymore. So we know how the editing works, but where does it actually happen? Well, the source material gives us two distinct pathways. In vivo therapy means the new genetic instructions are delivered directly into the patient's body. Ex vivo therapy, on the other hand, is a bit different. Here, cells are actually extracted from the patient, modified outside the body in a laboratory setting, and then those freshly corrected cells are reintroduced back into the patient. Section 3. Delivering the genetic payload. Now, what's really interesting about this next concept is how we actually get the edits into the cells. We use carriers called vectors to get the new genes inside the microscopic cells where they're needed. First, we have viral vectors. Think about this for a second. Viruses have spent millions of years evolving to be nature's absolute best microscopic hijackers. Scientists essentially hollow out the bad stuff, turn them into microscopic delivery trucks, and pack them with our therapeutic genes. We're talking things like adenovirus, retrovirus, or lentovirus. The advantage? Extremely high efficiency. The disadvantage? Your body's immune system might still see it as a threat and attack the truck. Alternatively, we have non-viral vectors. These use methods like liposomes, nanoparticles, or even direct DNA injection. The trade-off here is pretty clear. They are generally safer and carry a much lower risk of an immune response, but currently they just don't pack the same high-efficiency punch as the viral vectors do. All right, Section 4, curing the incurable. Just look at this wide array of applications. It's not just for rare genetic disorders like cystic fibrosis, hemophilia, or sickle cell disease, though, to be clear, it is completely revolutionary there. Gene therapy is highly versatile. In cancer treatment, it can actually activate the body's own immune system against tumors, or even introduce suicide genes directly into cancer cells to destroy them from the inside out. It's also being heavily explored for viral infections like HIV and hepatitis. It even holds massive potential for neurological disorders like Parkinson's and Alzheimer's, and for cardiovascular diseases, where it could realistically help repair damaged heart tissue or improve blood vessel growth. And honestly, that brings us right back to this incredibly powerful quote from our source material. The main goal is to treat diseases at their genetic root, rather than only managing symptoms. This is exactly what you should take away from this explainer. The profound advantage of this highly targeted approach is the potential for a long-term or even a permanent cure. Imagine a life where a patient with a rare genetic disorder is no longer dependent on lifelong daily medications, all because their underlying cellular blueprint has been permanently repaired. Section 5, the promise and peril. Look, as much as gene therapy sounds like literal magic, the reality is that we are still facing very real, very steep hurdles. The cost of these treatments can be astronomical, making them completely inaccessible to many people. We already mentioned the risk of immune reactions to viral vectors, but perhaps the most concerning thing from a medical standpoint is that editing DNA can sometimes have unintended genetic or off-target effects. Because this entire field is still relatively new, we simply have limited long-term safety data to know exactly how these edits might behave over decades. And finally, of course, there are the ethical concerns. The ethical issues here are profound. As the source outlines, germline therapy involves the modification of human embryos, which passes those edits down the human lineage. There are deep genetic privacy concerns regarding who actually holds this highly sensitive data. Then there's the very real question of accessibility and fairness. I mean, if only the ultra-wealthy can afford genetic cures, what does that do to society? And inevitably, the sources point to the risk of genetic enhancement, the so-called designer babies, where genes aren't being edited to cure disease, but to enhance human traits. The source material emphasizes that strict regulations are absolutely necessary to ensure safe and ethical use across all of these fronts. Which brings us to Section 6, the future of medicine. And this brilliantly illustrates what's coming next. The future is incredibly promising. We are rapidly moving toward genuine cures for inherited genetic disorders and highly advanced cancer treatments. But most excitingly, we are entering a true era of personalized genetic medicine, where gene therapy will be increasingly tailored to your specific individual genetic profile for the absolute best outcomes. We're also seeing the massive promise of combining gene therapy with stem cell technology, and ongoing research is fiercely focused on creating those safer, non-viral, highly efficient delivery systems we talked about earlier. Gene therapy is undeniably a revolutionary approach to medicine. By correcting defective genes, we have the genuine possibility of curing the previously untreatable. But as we gain the immense power to be the architects of our own genome, I want to leave you with this final provocative thought. As our CRISPR tools get sharper and our delivery vectors get safer, where exactly is the line between curing a disease and creating a designer baby? It is an absolutely massive responsibility, and it's a question we are all going to have to figure out together in the coming decades. Thank you so much for joining me in this explainer. I hope you found it as fascinating as I did.